Today we're going to talk about membranes. Membranes are hugely important. They define what a cell is capable of. They define how a cell will interact with its environment. So we have a membrane that surrounds the cell, that's the plasma membrane or cell membrane. And then we also, of course, have internal membranes. And those internal membranes are going to make up compartments, organelles, within the cell, and those compartments have specialized functions. So what you're seeing here is a diagram of the plasma membrane. We have a phospholipid bilayer, bi meaning two, so we have two layers of phospholipid. Remember that a phospholipid has a hydrophilic head, and then it has hydrophobic tails. The tails are fatty acids. Also, we've got some cholesterol molecules that are embedded into that phospholipid bilayer. You can see one here. The large purple structures are proteins that are embedded within the membrane. And in some cells, there can be more protein than lipid. On the outside of the cell, which is towards the top of this diagram, we have carbohydrates. You can see them here attached to fatty acids, and we can see them here attached to a protein. So that would be a glycoprotein. We also have proteoglycans. This is what you're seeing here, this long feathery molecule. So that's also a mixture of carbohydrate and protein. And it makes up what's called the extracellular matrix. The extracellular matrix is this protective layer around the outside of the cell. We don't have a cell wall. Instead, we have this extracellular matrix. And these proteoglycans make kind of like a jelly-like substance that helps protect the cell and also glues cells together. These large molecules that you see on the outside are collagen. And collagen forms part of the extracellular matrix and forms a major component of a lot of connective tissues, as we'll see later on. On the inside of the cell, we have these cable-like structures, which are part of the cytoskeleton. And the cytoskeleton is exactly like it sounds. It's a skeleton inside the cell that anchors the membrane to other internal components of the cell. Lipid molecules are hydrophobic, meaning that they don't mix with water. If you add lipid molecules to water, those molecules will come together. And there isn't really a force that's pulling them together. Instead, they're being excluded from the water. So every water molecule is trying to form hydrogen bonds with other water molecules. And as they do that, that pushes the fat molecules, the lipids, together. Now, once the lipids are together, they are large molecules, and there may be van der Waals forces involved at that point that help keep them together. Lipids share the common property of having a large region that is hydrophobic. So in some cases, the entire molecule may be hydrophobic. In other cases, we can have lipids where one end is charged and the other end is not, and that's an amphipathic molecule. So it has one end that's hydrophobic and one end that's hydrophilic. And you're seeing a few examples of lipids and pieces of lipids here. So a triglyceride consists of glycerol and then 3 fatty acids. The fatty acids, when they're free on their own, they do have a charged end, as you can see. And also the glycerol, when it's free on its own, it has some charges as well. But when we bring them together, we end up with a triglyceride molecule that is entirely hydrophobic. Some lipids contain cyclic structures, so ring structures. Those are known as sterols, and you can see an example down the bottom there, cholesterol. A free fatty acid is an amphipathic molecule because we have this hydroxyl group down here. And we have an uneven charge distribution between the oxygen and the hydrogen. However, when we take these fatty acids and use them to build lipids by attaching them to glycerol, we lose that hydroxyl. It's a dehydration synthesis reaction, and that hydroxyl is going to be pulled off. It's going to be combined with a hydrogen proton to make water. We also have this long region, usually referred to as the tail, that is nonpolar and very much hydrophobic. So again, even though glycerol and fatty acids are amphipathic in their free form, when they come together to build something like a triglyceride, we end up with a hydrophobic molecule. In the space-filling model here, the white is hydrogen, the black is carbon, the red is oxygen. And notice those long tails that contain only hydrogen and carbon. Remember that those two atoms have very similar electronegativities. That means that we don't have any uneven distribution of charge. All of those polar bonds are very, very good at storing potential energy. And we'll talk about that later when we talk about nutrition and so on. Sterols, like cholesterol, are part of a larger group known as steroids or steroid alcohols. They all share a few characteristics. They have these ring structures that I mentioned already, and you can see there's 4 of them. And then they also have a hydroxyl, which, as you know, is characteristic of alcohol. So we can see the hydroxyl here. And this is the fully formed molecule. This molecule may end up embedded within the membrane. We've got our hydroxyl group there, which is polar, and then the rest of this molecule is nonpolar. So that little tiny hydroxyl is the only charged part of the molecule, and it's sometimes considered to be the hydrophilic head of the molecule, even though it just consists of 2 atoms. So in the schematic diagram to the far right, that black dot is our hydrophilic head, our hydroxyl group. We've got the steroid rings, and then we've got a long chain that comes off of that that's kind of like a fatty acid. Again, the only part that is hydrophilic is that hydroxyl group. So within that hydroxyl of cholesterol, we have an uneven distribution of charge between hydrogen and oxygen, but we don't have a full-on charge. In phospholipids, we have a full-on negative charge and a full-on positive charge. So you can see our negative charge here on the oxygen of the phosphate, and you can see our positive charge here in the nitrogen of the choline. So that head has a full positive charge and a full negative charge, and that means that this hydrophilic head interacts with water very strongly. Fatty acids are nonpolar and hydrophobic. What you're seeing here is an electron micrograph, a photo taken with a transmission electron microscope of the plasma membrane. This is very high magnification. So you can't actually see the membrane with a light microscope. If you're looking at cells in the lab, you will see a dark line perhaps on the outside of the cell, but you're actually seeing the much thicker layer of proteins that make up the extracellular matrix. This is an incredibly thin structure. We've got 2 parts, 2 layers, and each layer is referred to as a leaflet. So we have a cytosolic leaflet or cytoplasmic leaflet on the bottom in that diagram. That would be in contact with the interior of the cell. And then we have an external leaflet on top of that. This is a very stable configuration. And that's because in this configuration, all of the hydrophilic heads, remember they're very strongly hydrophilic, are in contact with water. Well, the hydrophobic tails are not. They're excluded from the water. So that's a stable configuration. Also, we do have van der Waals forces between the tails that help maintain this structure. Remember that in any molecule, electrons are in constant motion, and they're moving very quickly. So at any one point in time, we have slightly uneven charges, but they don't last for very long. They just last for a tiny fraction of an instant. Well, if we have a large molecule, those tiny little attractive forces can be significant, and fatty acids are rather large molecules. Cell membrane, or plasma membrane, is selectively permeable. And what that means is some things can cross quite easily, and some things can't. And the cell is able to determine what gets in and what gets out. So in this diagram here, again, you're seeing the phospholipid bilayer. You're seeing some proteins that are crossing through the phospholipid bilayer or embedded partially into it. And you can see a rather interesting protein here that's got a channel in the middle. And it's channels like this that allow certain molecules to pass through and allow the membrane to be selective. Membranes do a lot of important stuff. So they are responsible for the selective uptake of ions and molecules, and also the selective release of substances. They play a role in cell compartmentalization, so they make up compartments like the Golgi body, the endoplasmic reticulum, etc. And because these compartments are isolated from the rest of the cell, we can have specialized functions occurring just in that little compartment. We can have a different pH in that compartment. We can have different enzymes present in that compartment. They're involved in protein sorting. So we'll talk about this later, but proteins that are transported into the rough endoplasmic reticulum can end up embedded in a membrane, like the plasma membrane, or they can be excreted. If a protein doesn't make its way into the endoplasmic reticulum, it's going to remain in the cytosol, in the liquid portion of the cell between the organelles. Energy production. So the mitochondria is essentially a battery. There's two membranes, and what the mitochondria does is it builds up a strong concentration gradient of hydrogen protons across the inner membrane. And then the protons run through something called ATP synthase, and the gradient of charge is used to manufacture ATP. And we'll talk about that later too. Cell signaling. So cells have proteins attached to them that can be receptors for hormones, for neurotransmitters. They also have carbohydrates exposed on their surface that can identify them, as we've talked about. Cell division. So animal cells, like our cells, will grow in size as they're getting ready to divide. And when they do divide, of course, the cell is just going to pinch off into two. And that's accomplished by moving the membrane. There are motor proteins associated with the underside of the membrane. Those motor proteins can change shape, and they can change the shape of the cell. Adhesion to other cells and to the extracellular matrix. So the membrane forms an outer layer of goo, protein and some carbohydrate, known as the extracellular matrix, and it also contains a lot of proteins that are used to anchor cells together. All membranes are made up of phospholipid and some protein, but there can be a huge variation in the amounts. Carbohydrates are quite often present, but not always, and when they are present, they're generally present only on the outside of the cell. The proportions of protein and lipid in a membrane can tell us quite a lot about what the membrane does. So let's just take a moment and compare a few different membranes. And we'll start with the plasma membrane of a red blood cell. Red blood cells are essentially bags of hemoglobin, and the hemoglobin is used to transport oxygen. But they're rather unspecialized. There's not a whole lot in the way of metabolic activity going on in the cell, at least not compared to a lot of other cells. And they have a rather unspecialized structure to their membrane. They have a pretty much even mix of protein and lipid. Now, if we compare that to the inner membrane of the mitochondria, the situation is quite different. So mitochondria have two membranes. They have an outer membrane and an inner membrane. And the inner membrane is 3/4 protein. Remember that proteins do all the work in a cell. These proteins consist of channels and pumps and other things we'll talk about eventually when we talk about cellular respiration. But there's a lot of activity inside the mitochondria, and the membrane serves as kind of a scaffold to hold all of these important channels and enzymes in place. In fact, in many organelles, we can have enzymes lined up like an assembly line, and they're all held in a specific order so that a biochemical pathway can occur along that membrane. Now, if we compare this to a nerve cell, and specifically looking at the axon of the nerve cell, that's the long tube that comes off of a nerve cell that conducts an action potential, it's mostly lipid. So it's about 3/4 lipid. And in fact, in myelinated nerve cells, we have many, many layers of plasma membrane wrapped around the outside of that nerve cell. The lipid acts as sort of an insulator. So there's not a lot of protein in these regions because the cell is not going to be moving ions across the membrane in these myelinated areas. So instead we have lots of lipid that will insulate the cell and prevent the movement, the easy movement of materials across the membrane. So at this point, you understand the basic structure of the membrane. What I want to do is just going to go back and talk a little bit about the history of how we worked out this structure. And this is not a history course, so I'm not going to expect you to remember names and dates, but I just think it's kind of interesting for you to gain an appreciation of how this knowledge was collected. Okay, so again, we have a typical phospholipid here. It's got a hydrophilic head, which contains a choline and a phosphate, and then an uncharged glycerol. And then we have hydrophobic fatty acids attached to that. Note that the fatty acids can contain double bonds. And remember, when we have a double bond, that fatty acid is said to be unsaturated, and it causes the chain of carbons to kink slightly. We've also discussed how when you take phospholipids and throw them in water, they're going to try to form a stable configuration. The easiest thing for them to do is to simply form spheres. So they'll form spheres with the hydrophilic heads around the outside and the hydrophobic tails on the inside. But if you take phospholipids and you put them in a thin layer on top of a water bath and you shake it, you can actually get them to form a phospholipid bilayer. And this is something that's been known for well over 100 years. So again, we know today membranes are made up of phospholipid bilayers. The interior is hydrophobic, and the hydrophilic heads interact very strongly with water. As early as the 1880s and 1890s, it was realized that lipids were a major component of the plasma membrane. Later, researchers discovered that proteins were also quite important, but it took a while for people to work out how those different bits and pieces were put together. So in 1895, a guy by the name of Overton did some experiments where he collected plasma membrane. It's not that difficult actually to collect up red blood cells, split them open, rupture them, and then collect the membrane as a layer on the surface of a water bath. And that's what he did, and that's what subsequent researchers did to collect these membranes. Now, again, this is not a history course, so I don't expect you to know these names or dates. In 1917, another researcher proposed that the membrane was a single layer of phospholipids. So we're halfway there. Not that long after that, it was hypothesized that the membrane was made up of two layers. It was a lipid bilayer. And this made sense. This was a stable configuration that wouldn't take a whole lot of energy on the part of the cell to maintain. Now after this, people started thinking about how proteins fit into this model because they were realizing that proteins were also associated with the membrane. Some of the earlier models suggested that the proteins were stuck to the hydrophilic heads. Now, this didn't quite work because that's not all that stable. If the proteins were just held there by ionic bonds, for instance, they could be displaced quite easily. When the electron microscope came into widespread use, people were able to look at the plasma membrane and see those two dark lines that I showed you in that photograph earlier. And this was something they saw in all membranes. And they started referring to this as the unit membrane. But again, people weren't quite sure what those dark lines were. In 1973, Singer and Nicholson came up with a model that pleased everyone, and it made a lot of sense. So they were able to show that the proteins and phospholipids and so on that people had collected from these membranes fit together with the proteins embedded into the membrane. And there are some peripheral proteins as well that are attached to the outside. This image here shows two plasma membranes. And again, we can see those two dark lines that represent the hydrophilic heads. And we can also see the space in between the cells. And that intercellular space would be filled up with proteins of the extracellular matrix. And they act as a glue to hold the cells together. This is our current model of biological membranes. And it's held up quite well over the last 50 years. Lots of experiments have supported that this is, in reality, how things are arranged in the membrane. It's known as the fluid mosaic model. It's a mosaic because it consists of more than one part. So it consists of proteins and lipids. That's the mosaic part of the title. And it's fluid, meaning that things are moving around. So those proteins can move from side to side, and those phospholipids can move from side to side. In fact, Singer and Nicholson famously refer to the proteins as icebergs floating in a sea of lipids. And that really is what's going on. It's a very dynamic, very fluid structure. And unlike in a lot of the earlier models, this model suggests that the majority of proteins that are associated with the membrane cross the membrane, or they're embedded very deeply into one of the two leaflets. If you were to take the two leaflets and separate them, the protein would generally stick to one leaflet or the other, as you're seeing in this diagram here. And this has actually been accomplished. You can take cells and freeze them very rapidly into ice, and then whack those frozen cells with a very, very sharp blade. And if you're lucky, some of those cells will split between the two leaflets. So the membrane will be split into two leaflets. And then you can take those cells with the ruptured membrane and take a look at them under an electron microscope. So there were a number of experiments where people did this, and when they looked at the split leaflets under an electron microscope, they were able to see proteins embedded into the membrane. So on the top here, we've got the view of the inside of the extracellular layer. And again, this is with a scanning electron microscope, so it gives us 3D images of the texture of the surface we're looking at. And this is the inside of the cytoplasmic layer. Notice that it's a lot rougher. All of those bumps are proteins. And when you rupture a membrane between the two leaflets, proteins that cross the entire membrane tend to stick to the cytoplasmic layer. And the reason is that the cytoskeleton is down here. So we have fibers that make up the cytoskeleton, and these proteins are attached to those fibers. So the proteins tend to stay on the cytoplasmic leaflet. That first experiment, the freeze fracture experiment, demonstrated that the membrane is a mosaic. So we've got protein and lipid, but the proteins aren't just stuck loosely to the outside, they're an integral part of the membrane. They're embedded within the membrane. Other experiments showed that the membrane is also fluid, and we'll have a look at a couple of those. So it's possible to take two cells and fuse them together to create a hybrid cell. What you do is you take your two cells, bring them close together so they're touching, zap them with electricity. That causes little holes to open up in the cells so that they can fuse into one big cell. In this experiment, what researchers did was they manufactured antibodies that would recognize a protein or proteins on a mouse cell, but not recognize the same or similar proteins on a human cell. And they also were able to create antibodies that would bind to proteins in a human cell, but not bind to proteins on the mouse cell. It's possible to bind dye molecules to antibodies. So what you can do is you can dye your mouse cells one color, dye your human cells a different color, and the dye molecule is bound to an antibody that's bound to integral proteins in the membrane. So you take your antibodies, you throw them in with the cell, you let the antibodies bind, and then you wash off the excess, and then you bring the two cells together to create your hybrid. Immediately after doing this, you'll see one big cell that is half one color and half another color. But if you leave it for an hour, it becomes one color. What's happening is those proteins are moving around randomly within the membrane. They're dispersing themselves, and we don't see those two colors anymore. We see the combination of the two colors throughout the cell. This next experiment is a variant of that. So imagine we take a cell and we're going to make a fluorescent dye, and that dye is going to be bound to an antibody, and that antibody is again going to bind to proteins that are embedded in the membrane. So we have our cell and the proteins have been dyed, and now we're going to take that cell and put it under a laser. So a laser is a very fine, very straight, very intense beam of light, and we can use that to destroy the dye molecules within a small area. So we're going to bleach that area. It's going to be clear after we hit the membrane with the laser. The laser is only going to destroy those dye molecules, though it's not going to disrupt the membrane. If we leave our cell and watch it under the microscope, what you'll see is color will start to come into that bleached area. And it's because the dye molecules attached to proteins are moving into that region. So the membrane is fluid, it is dynamic, but I should point out that the movements we're talking about are almost entirely lateral movements. It's very uncommon for a phospholipid to jump from one leaflet to another. Instead, they're moving sideways or laterally within their leaflet. There are two broad categories of membrane proteins. We have integral proteins that are inserted into the membrane and quite often span the entire membrane. The defining feature of integral proteins is they have at least part of their structure embedded into the hydrophobic core. So in some cases, most of the protein is on the outside, but there's an anchor that goes into the membrane. And then we have peripheral proteins which are attached to the surface, and they're fairly loosely bound. They're held in place by ionic bonding and other rather weak bonds. And in some cases, they may come and go. This is an example of an integral protein. It's also an example of a transmembrane protein because it crosses the membrane. So you can see there's parts that stick out on the exterior and parts that stick out on the interior of the cell. Very commonly, integral proteins will be anchored by an alpha helix. And you can see this one has several alpha helices that cross the membrane and embed themselves into the hydrophobic core. But not all integral proteins are transmembrane proteins. And here's an example here from Staphylococcus aureus. The vast majority of the protein is on the outside of the cell, but it's not loosely held to the membrane. Instead, there is one alpha helix that makes up part of that protein that is strongly embedded in the membrane. Alpha helices are used as anchors because they're largely hydrophobic. And you might be wondering, well, isn't there a lot of hydrogen bonding in the alpha helice? That's what holds its structure together, right? Well, the hydrogen bonding that creates this helix, as you can see in the diagram on the left, is between atoms that make up the backbone of the amino acid. But the majority of the functional groups that are found on amino acids within an alpha helix are hydrophobic. They're nonpolar. In the diagram on the right, you're seeing the sequence of a typical alpha helix that might be embedded into a membrane. The green amino acids are charged, or they have a slight charge. They're polar. And the yellow amino acids are hydrophobic. And notice that the charged amino acids, the green ones, are found towards the ends of the alpha helix, the part that is going to interact with the hydrophilic heads. So those amino acids, threonine, serine, etc., that are polar are going to interact with the hydrophilic heads, and that hydrophobic region in the middle is not. So this is a very, very stable situation. It's rather difficult to displace that structure. If you tried to push it through the membrane, the interactions between the polar amino acids and the hydrophilic heads would resist that movement. Again, the majority of the amino acids in that alpha helix are going to be nonpolar, the ones shown in yellow here. And we're going to have some polar amino acids as well, but they're at the ends of the alpha helix. Those are the parts that will interact with the hydrophilic heads of the phospholipids. Here's an example of a rather typical transmembrane protein. This is something known as Glycophorin A. You don't need to worry about that, but this is a protein that is found in the plasma membrane of red blood cells, and it transports glucose into the cells. Notice that we've got two molecules there. So this is a protein that's made up of two parts. And for both of those parts, there's an alpha helix that embeds itself into the hydrophobic core of the membrane. This diagram shows some of the several ways that proteins can attach to the membrane. So on the far left, we have a peripheral protein that's attached to the cytoplasmic side of the membrane. It's going to be loosely attached. Next to that, we have a single pass transmembrane protein. Single pass refers to the fact that it only passes through the membrane once. It's got one alpha helix that's going to anchor it into the membrane. Next to that, we've got a peripheral protein that is more strongly attached to the membrane. It's covalently bound to a phospholipid. And finally, on the far right, we've got a multiple pass transmembrane protein. Multiple pass because it crosses the membrane several times. Every time it crosses, there is an alpha helix that anchors it into the membrane. And the more passes there are, the more alpha helices, the more strongly embedded into the membrane it will be. The proteins that are embedded in the membrane can do a number of important things. We have some proteins that are involved in transport. So in the example there in part A, you've got a simple channel, and the opening in that channel is the right size, has the right charge and the right characteristics to allow a certain molecule to move passively down its concentration gradient. Next to that, we've got a pump that requires ATP to move materials across the membrane, quite often against their concentration gradient, so from low to high. So in this case, the pump is a molecule that will grab onto something and change shape if it's supplied with ATP and dump that material on the other side of the membrane. Enzymes are embedded in membranes. And this is really important in the mitochondria. As I mentioned, the inner membrane of the mitochondria has lots of channels and also lots of enzymes embedded within it. So here we've got an example of a biochemical pathway. So we have a triangle, whatever that might be, converted into a square, converted into a circle. So this is an enzyme catalyzed series of reactions. And the enzymes that are working together in this process are located next to each other in the membrane. So they can all be stuck right next to each other, forming an assembly line. Signal transduction. So here we have a signaling molecule, could be a hormone, for instance, like insulin, and it's going to bind to a receptor, and the binding site is on the outside of the cell. After that signaling molecule binds, this protein is going to change shape, and it's going to catalyze some sort of reaction on the inside of the cell. So it's relaying a signal from the outside to the inside of the membrane. Glycoproteins can play a role in cell-to-cell recognition. In this example here, the cell on the bottom has a glycoprotein embedded in the membrane. The sugar part of the glycoprotein is exposed to the external environment, and another cell with a protein that has a receptor site for that specific sequence of sugars will recognize that glycoprotein, and then these cells may bond or any number of other things might happen. We have proteins that join cells together. So in that example there, we've got two integral proteins that have bonded to each other to hold the cells together. And finally here, we've got attachment to the cytoskeleton and also to the extracellular matrix. So the bottom of that diagram is the interior of the cell. We've got proteins that are bound to the cytoskeleton. And those same proteins on the other side, on the exterior, are interacting with proteoglycans and also collagen fibers, which make up the extracellular matrix. And here's another example of glycoproteins and also glycolipids. So we can have carbohydrates attached to the outside of the cell, attached to proteins, or attached to lipids. Attaching sugars to something is a process known as glycosylation. So we would say that these lipids and proteins have become glycosylated. The other thing to note is that really feathery structure that you see mixed in with the collagen, that would be a proteoglycan. It's a mixture of protein and sugar, but in this case, it's mostly sugar. These carbohydrates are very important when it comes to embryo formation. In the early embryo, we have some populations of cells that wander around quite a bit. They wander through the embryo, and they know where they are in the body based on the carbohydrates that are exposed on the surface of the surrounding cells. If a cell finds itself in tissue that's developing into the liver, it will become a liver cell. We have to consider these carbohydrates when we're doing transplants, tissue or organ transplants. The carbohydrates that you have on your cells will differ from the carbohydrates that other people have, and your body may reject that tissue. It'll recognize it as being foreign. The immune system is aware of these carbohydrates, and if it sees cells, if it comes across cells that don't share your particular pattern of carbohydrates, it may elicit a response and attack that tissue. Blood types, A, B, O, etc., are the result of carbohydrates that are found on the cell membrane of red blood cells. And if you look at that top diagram, note that whether you have O, A, or B blood type, you have this common sequence of carbohydrates attached to a particular glycolipid. However, if you have type A blood, you also have this carbohydrate stuck onto that core sequence. If you have B, you have this one. If you have AB blood, you have both of those two types of glycolipids. And there are lots of other blood types. That's what you're seeing on the bottom diagram there. Don't worry about the names of all of these, of course, but just to show you that there are many different glycoproteins and glycolipids that determine blood typing. In order for membranes to work properly, they need to be fluid, but not too fluid. So we want the membrane to stick together and not fall apart, of course, but we don't want it to be too rigid. If it's too rigid, then there's a chance that the membrane will break or will fracture in some way. There are three main ways that the membrane fluidity can be regulated. The first important one is the length of the fatty acids. If the fatty acids are longer, then we have more opportunity for van der Waals attractions, and the membrane becomes a bit more rigid because of that. And some cells that require more fluidity may have shorter chained fatty acids in their membranes. Whether or not the fatty acids are saturated or unsaturated is also very important. Remember that an unsaturated fatty acid doesn't have as many hydrogens as it could. It's not saturated with hydrogens. And that's because there's double bonding in the carbon chain. Double bonds introduce a kink. Kinks in the unsaturated fatty acids prevent them from packing close together. So it'll stop the phospholipids from packing as closely together as they could, and that means that the membrane will remain more fluid. There are a number of fish, for instance, that can survive very, very cold water by introducing more unsaturated fatty acids into their phospholipids to keep their membrane fluid at cool temperatures, because otherwise it would get somewhat rigid. And you've probably experienced this yourself in your kitchen. Your vegetable oil, it contains unsaturated fatty acids and it remains fluid at room temperature, whereas the butter you have in your kitchen contains saturated fatty acids. They pack together and we get a solid. And finally, cholesterol can have a big impact on the fluidity of a membrane. Your cells are constantly adjusting the stability of the membrane, and one of the main ways is by adding or removing cholesterol. So you can see that cholesterol fits itself in between the phospholipids. We have the hydroxyl group that is going to interact with the polar heads of the phospholipids, and then we have those ring structures that are hydrophobic that will interact with the fatty acid tails through van der Waals forces. What this does is it introduces a little space between the phospholipids, and at low temperatures that increases the fluidity of the membrane. At high temperatures, however, the extra bonding, the extra van der Waals forces, help hold the phospholipids together so they're not just moving all over the place. So the effect of cholesterol in general is to stabilize the fluidity of the membrane and keep it about the same despite temperature changes. Okay, I've mentioned that membranes are semi-permeable. Let's have a look at that. So biological membranes are of course semi-permeable, but we can also make artificial membranes that are semi-permeable. What we've got here is a simple lab setup where we've got two compartments filled with water or another fluid, and we've got an artificial lipid bilayer separating those two compartments. Now what would happen if we added a solute to one side? And I should probably define what a solute is. A solute is something that you dissolve in a solvent. The solvent is the fluid that does the dissolving. So if we added salt to water, salt would be the solute and the solvent would be the water. So if we add our solute here, we can do little tests to see whether or not it can cross the membrane, and we can modify our artificial membrane to see if we can help it out. So let's say that we added phospholipids to our membrane that had a lot of unsaturated fatty acids. If that's the case, the unsaturated fatty acids would make the membrane more fluid, which would make it leakier. We could add proteins, and perhaps those proteins would act as channels and allow the solute to move more easily. And what we find with these kinds of experiments is that even without any proteins, lipid bilayers are selectively permeable. They allow some things to pass through, but other things cannot. Very small molecules can move across the membrane freely, assuming they're not charged. So oxygen, nitrogen, carbon dioxide, they can sneak through the tiny spaces between the phospholipids. They have an even easier time if the phospholipids contain kinked, unsaturated fatty acids. However, really small molecules and even individual atoms can't get across the membrane easily if they're charged. And that's because they're going to interact with the hydrophilic heads of the phospholipids. So a chloride ion, for instance, is negatively charged, and it is going to stick to the positively charged choline. Sodium is positively charged. It's going to stick to the negatively charged phosphate of the hydrophilic head. Larger molecules, some can pass through, some can't, some need some help. It depends on the shape of the molecule, and again, it depends on the charge as well. Molecules that are hydrophobic, like other lipids, even if they're fairly large, can get across the membrane without too much trouble, and that's because they don't interact with the phospholipids. Now, they may actually get stuck in the hydrophobic core in the interior, but they're not going to interact with the hydrophilic heads. So hydrophobic molecules, even if they're fairly large, can get through easily. Uncharged small molecules can get through very easily. Small molecules that are not charged but are polar have a bit more difficulty. So water, for instance. Water will move across the membrane, but your cells have a transporter, an aquaporin it's called, that allows water to move much faster. These little pores allow the water to move about 10 times faster. Large, uncharged polar molecules, if they're uncharged, that's going to help them get across, but because they're large, they're going to have difficulty moving the phospholipids aside to go across the membrane. And ions, even though we're talking about individual atoms here. Remember, an ion is a charged atom. Even though we're talking about individual atoms, because they have a full charge, they don't get across the membrane very easily at all. So the cell has to have channels and pumps and so on to move these things effectively. If we want to make a membrane with high permeability, the easiest way to do that would be to use fatty acids that are short and also unsaturated. Cholesterol affects the permeability as well. So adding cholesterol to a membrane, again, makes it more stable, but it tends to make the membrane a bit more rigid. It maintains the structural integrity of the membrane, but at the cost of permeability. So permeability is lowered when we add cholesterol. Cholesterol, however, does play a critical role in maintaining the structure, maintaining the health of membranes. We tend to think of cholesterol as being bad for us, and it is if you've got too much of it and the wrong types, but we do need cholesterol for this important role. All right, so how do molecules move across membranes? The simplest way is just simple diffusion. So diffusion is the movement of molecules down a concentration gradient from an area of high concentration to an area of low concentration. And incidentally, when you see square brackets like that, it's a shorthand for concentration. You'll have seen that before if you've taken any chemistry. So if the membrane is permeable to the molecule can move down its concentration gradient. Diffusion is a passive process. What I mean by that is it doesn't require any energy. Diffusion is simply due to random motions and collisions. So the molecules in our little setup here were all concentrated on one side. We've got two different solutes here. But if we leave this to equilibrate, it will eventually, and that's just due to random collisions between molecules. We'll get to a point where we have the same concentration of both solutes on either side, again assuming they can move freely across the membrane. And at that point we will have continued random movements of stuff, but it will happen at the same rate in both directions. So if we put dye molecules in a permeable sac, let's say, and we put that into water, provided the dye can move across the membrane, the dye molecules will bounce into each other, they'll spread themselves out until we have this random dispersion, and we have equilibrium being reached between our membranous sac and between the water that's surrounding it. Osmosis is a special type of diffusion. It refers specifically to the movement of water across a semi-permeable membrane. And the water is moving because there's solutes that can't get across the membrane. So let's say that we have a solution that is 5% salt. That means that it's 95% water. Now let's say we put that solution on one side of a membrane, and on the other side of the membrane we have a solution that's 10% salt. That means that solution would be 90% water. Let's say that the salt can't get across the membrane, but the water can. The water will flow down its concentration gradient. It will go from the 95% water solution down to the 90% water solution. And that is osmosis. So here we have our setup. And we have a membrane that's permeable to water, but it's not permeable to the solute, whatever that might be. So the solute is represented by these little green dots. And you can see we've got more solute on one side than the other. That solute is taking up space that could be occupied by water. So the concentration of the water is higher on the left than it is on the right. And the water is going to move down its concentration gradient. What's going to happen is the water is going to move across towards the right, and it's going to actually raise the level of the water in that container. We'll get to a point where the force of osmosis moving to the right is equal to the force of gravity pushing down. And this is how an osmometer works. and we'll get to that in a moment. Now notice that most of the water is moving to the right, but it is a random process, so there will be some water moving back as well. But we will get to a point where we have an equilibrium. And again, the equilibrium is going to be established by the point where the force of osmosis is equal to the force of gravity pushing down on the right-hand side. So in a case where gravity is involved like this, we'll never actually get equal concentrations on both sides. There are three important terms here that you need to know. And these terms are used when we're comparing the concentration of different solutions. Let's say I have a 5% solution of salt and I have a 10% solution of salt. The 5% solution is hypotonic to the 10% solution. Hypo means less than. The 10% solution is hypertonic to the 5% solution. Hyper means more. So think of being hyperactive, you're more active than you should be. Think of having hypothermia, you're cooler than you should be. Isotonic means the same. So a 5% solution of salt is isotonic to a 5% solution of salt. Now we could be talking about different solutes that won't move across the membrane. So I could say a 5% solution of salt is isotonic to a 5% solution of sugar, if we're using a membrane that is impermeable to both salt and sugar. As far as the water is concerned, the water will move down its concentration gradient. The water doesn't care whether we're dealing with salt or sugar or a mixture of the two. Now, when we're using these terms with respect to cells, we use them in a very specific way. So if we say that a cell is in a hypertonic solution, that means there's more solute outside the cell than inside the cell. If we say that a cell is in a hypotonic solution, that means there's less solute outside the cell than there is inside the cell. And you can predict, of course, which way the water is going to move. The water is always going to move down its concentration gradient. The important thing to note is that the water gradient is opposite to the solute gradient. So in this example here, let's say that the left-hand side of the membrane is the interior of the cell, and the right-hand side is the exterior. The green represents a solute of some sort, salt for instance. We can see that there's more solute outside the cell than there is inside the cell. So this would be a cell that's in a hypertonic situation. The solute can't move across the membrane. If that's the case, the water is going to move to try to equalize the water concentration. And the water is going to move across from the left to the right. So here we've got a little sac that we've made out of an artificial semi-permeable membrane, like dialysis tubing. So we use dialysis tubing in the lab all the time to concentrate solutions and so on. And we use it for simple little experiments like you see here. So imagine we've got our little semi-permeable sac, and this sac is permeable to water, but it's not permeable to sucrose. So the sucrose can't go across that membrane, but the water can. If we take a 2% sucrose solution and we put that into distilled water, so pure water, the water is going to move in. And again, the reason for that is because inside the bag, it's 98% water, but outside the bag, it's 100% water. So the water is going to move down its concentration gradient to basically dilute the sucrose that's inside the bag. Now, if we were to throw that into a 2% sucrose solution, we've got an isotonic condition. And in an isotonic condition, we do have water moving back and forth because, again, the motion is just random. It's just diffusion, but the rate in and out is the same. Now, if we took that 2% sucrose solution and threw it into a hypotonic condition, we've got more sugar outside, we've got less water outside. So we've got 98% water in the bag, 90% water outside the bag. The water is going to leave the bag. So the net movement of water will be out of the bag and the bag will shrink. An osmometer is a rather common piece of lab equipment, and it's used to measure concentration. It's actually used to compare the concentration of two solutions. So you've got one solution in the machine that you know the concentration of. You've got another one that you want to test, and you test it by basically comparing the solute content of the two. So this is quite simplified, this little diagram here, but this is the basic principle. Essentially, you've got a solution that has a lot of solute on one side, and then you've got a solution on the other side that has less solute. So in our example here, the hypotonic solution on the left contains more water because there's more space for water to exist. The solution on the right, the hypertonic solution, contains less water because space that could be occupied by water is being occupied by other molecules instead. And the water is going to flow across that membrane, so this membrane is impermeable to the solutes, to try to equalize the concentration. That will cause the water to rise, and as I mentioned in the example before, it'll get to a point where the force of gravity pushing down is the same as the force of osmosis pushing across the membrane. And this will give us the concentration or the osmotic pressure of the solution. How do these different situations affect the volume of a cell? If we have a cell in an isotonic solution, that means we have the same solute concentration inside as outside. And although water is going to be diffusing back and forth, we're not going to see a change in the volume of the cell. Your cells are happiest in this state. So the interstitial fluid, the fluid that is found between your cells, ideally should have about the same concentration of solutes as what's inside your cell. Now that's not always the case because of course your cells are trying to concentrate things and they're trying to get rid of things, but it's close to an isotonic situation. The interstitial fluid comes from your cardiovascular system. So it's basically plasma that seeps out from capillaries. A lot of that fluid is reabsorbed and goes back to the heart, but some of it doesn't. Some of it stays in between the cells. The concentration of solutes in your blood has to be closely monitored and controlled by your body to maintain this close to isotonic situation. Now let's say that we took a cell and we put it in a hypertonic solution. That means there's more solute or less water outside the cell compared to inside. Water will leave and the cell will shrink. If we do the opposite thing and we put a cell into a hypotonic solution, that means there's more water outside because there's less solute outside. Water will move in and the cell may rupture. Now that's of course if the cell doesn't do anything. Keep in mind that cells do have pumps. They can deal with this by pumping out excess water. But for a cell, an animal cell particularly, to be in a hypotonic solution for an extended period of time, usually not a good thing. Plants, on the other hand, prefer to be in a hypotonic solution. So the top diagram here shows what would happen to an animal cell, like a red blood cell. If we put it in a hypotonic solution, then the cell may lyse, which is a fancy way of saying rupture. So water rushes in, the cell expands, the membrane expands. The metabolic rate of red blood cells is not that great. They don't do a whole lot to maintain themselves, so there's a good chance that the cell will pop. If it's in an isotonic solution, then the cell is going to stay the same, and if it's in a hypertonic solution, the cell is going to shrivel, which is going to be detrimental to the cell as well. We need water inside the cell, of course, for all the chemical reactions of life to occur. Plants and also bacteria are quite different because they have a cell wall. So let's start with the isotonic situation. What's happening is the cell is going to be flaccid. Water is going to move in and out, but you can see it's quite different from the turgid condition. The turgid condition is when the plant cell is in hypotonic solution. So in this case, the water rushes in, but the cell wall pushes back. And we get this equilibrium forming where the force of water rushing in is the same as the force of the cell wall pushing back. So this is like a kind of inflated balloon, I guess you could say. And plants maintain structure by having these turgid, rigid cells. If a plant isn't kept in a hypotonic solution, so if the water in the soil is isotonic to the cells of the plant actually wilts. It becomes flaccid. So plants prefer to be in a hypotonic environment. If a plant is placed into a hypertonic solution, what happens is the water leaves and the membrane pulls away from the cell wall, and quite often it ruptures. Now, we're not talking about plants, so I don't expect you to know too much about what goes on in plants, but next term we will be looking at microbiology, and this is also what occurs in bacterial cells. They have a cell wall, and usually that cell wall is under a lot of pressure. They're turgid. The water is pushing against the cell wall. And if you can weaken the cell wall, which a lot of antibiotics do, that can cause the bacteria to rupture. So that's kind of an easy target for killing bacteria. Weaken the cell wall and they will pop in hypotonic conditions. And this is what a plant might look like if it's in isotonic conditions. So plants do prefer to have these inflated under pressure cells because they give them support. So far, we've been talking about the passive movement of materials across a membrane without any form of assistance. But that's not how things generally happen in cells. Cells use integral proteins to move materials into the cell that the cell needs to concentrate, and also to get rid of waste and secrete materials. These proteins can be channels. This is a protein that has an opening in the middle that allows certain substances to move down their concentration gradient. We can be talking about transporter proteins or carrier proteins. These are proteins that will grab onto large molecules. The protein changes shape and moves that material to the other side of the cell. And then we have pumps. Pumps are using ATP as a source of energy to pump materials across the membrane. And quite often they're pumping materials against their concentration gradients. Passive transport does not require any input of energy. Molecules are going to move down their concentration gradient. We can't move them against their concentration gradient unless we expend energy. But in passive transport, there's no expenditure of energy. Generally, we have a channel that sets up ideal conditions for a particular molecule to move across the membrane. Active transport requires energy. So with active transport, we can move things that would be very difficult to get across the membrane otherwise, like ions, for instance. And also we can move molecules up their concentration gradient. Here's an interesting example of a molecule that allows for passive transport, also known as facilitated diffusion. This is a large molecule that is produced by bacteria. And the bacteria species that produces this uses it to fight and kill competing bacteria. So it's a large molecule, as you can see. There's a hydrophilic interior that forms the channel, and then there's a hydrophobic exterior that can embed itself into the hydrophobic core of a plasma membrane. So what this cell does is it produces this channel, and then it inserts that channel into the cell membrane of a competing bacteria cell. And what happens is that channel allows water to leave the cell very quickly. So bacteria cells, as I mentioned before, they're usually under pressure. The water is under quite a bit of pressure inside, and the water can leave through this channel, and that's going to desiccate or dry out the bacteria cell and kill it. And we've made use of this particular compound and other compounds like it. It's something that's found in some ointments and antimicrobial creams. Aquaporins are found in many cell membranes. And what aquaporins do is they allow water to move more quickly across the membrane. So without the aquaporins, we would have some movement of water. Water would sneak its way between the phospholipids. But remember that water is a polar molecule. So that does impede its movement a bit. But with this channel, water can move much quicker. It can move about 10 times as quickly. Some channels can be closed and opened. and these are known as gated channels. So here we've got a potassium channel that can allow potassium ions to move down their concentration gradient. It doesn't require any energy to do that. The channel, though, is only open under certain circumstances. So there has to be a certain potential charge differential across the membrane in order for this voltage-gated channel to open. And we're going to talk a lot more about that when we get to the nervous system. So to summarize passive transport, it's a facilitated form of transport. It doesn't require energy, but exacting conditions have to be set up. Every channel is going to be differently shaped. It's going to have different charge distributions and so on, so that a specific molecule can move through that channel. And quite often the molecule can move in either direction, but it's always going to move down its concentration gradient. These can be regulated. So as I mentioned, they can be gated, which just means they can be closed and opened. Transporter or carrier proteins carry very specific things. They bind to a specific molecule, so it has to have the right shape, and then this transporter protein will change shape and dump that material on the other side of the cell membrane. Typically, they're only moving things in one direction. These are really important for getting metabolites into the cell, so getting amino acids and glucose and so on from the interstitial fluid into a cell that requires those things. So facilitated diffusion involves channel proteins, but it can also involve carrier proteins. Channel proteins are always going to transport things down their concentration gradient. This is always going to be an example of facilitated diffusion. Carrier proteins, on the other hand, some of them engage in facilitated diffusion, and some of them do actually require energy, and in that case it would be an example of active transport. Some examples of these proteins, the channels are quite often called porins. Think about the fact that they have a pore down the middle. Here's an example of one that conducts sucrose. Lactose permease is a large carrier protein that's found in the plasma membrane of bacteria, like E. coli. So if you have E. coli in your intestine, and you drink some milk that contains the sugar lactose, this permease molecule is going to become active, and it works just like the little diagram there in part B, where the molecule is going to grab onto lactose and the arrow is pointing at lactose. Very specific shape that the molecule has to have to fit into that permease molecule, and then this molecule, which is embedded in the membrane, is going to change shape and dump that lactose inside the cell. There are other transporters that can move things out of cells as well. Active transport requires an energy source of some type. Quite often that's ATP. Proteins that are actively pumping things across a membrane are known as ATP pumps. So the pump is capable of pumping molecules against a concentration gradient, so concentrating them inside the cell, for instance. Also, it's capable of moving things that would be very difficult to move otherwise. So ions do not move across a membrane readily. And quite often in cells, there's a need to set up a concentration gradient, to build a concentration gradient and move ions against their concentration gradient. So to concentrate ions between the two membranes of the mitochondria, for instance, or to concentrate ions inside a nerve cell. And that does require quite a bit of energy. So once again, we've got passive transport where there's simple diffusion of a substance across a membrane with no energy investment. Facilitated transport is a special case of passive transport. So it's transport using a channel protein, or in some cases, a transporter protein. And then we have active transport, which is going to require energy and will allow us to move things against their concentration gradient, which is quite often what needs to be done. Nerve cells need to set up a charge differential across their membranes in order to conduct an electrical signal. And there's several pumps that are involved in that. The pumps are moving charged ions across the membrane. One of the most important pumps is the sodium-potassium pump. So you can see that this is a pump that can move sodium in one direction. It moves it from the inside of the cell to the outside. And it can move potassium in the opposite direction. So starting with number 1 there, we've got our pump, and it's going to bind some sodium. So 3 sodium ions. ATP comes along, a phosphate is cleaved off and stuck to the pump, and that changes the conformation or shape of the pump. And the sodium ions are going to be moved to the outside and dumped outside. Next, we have potassium binding to specific spots inside that pump. We have the phosphate being pulled off, and with that comes a release of energy, and that energy is used to reconform the protein back to its original shape. And in the process, those potassiums get taken into the cell and dumped inside the cell. Because ions have a charge, when we move them and concentrate them on one side of a membrane, we also set up a voltage across the membrane. We set up what's called a membrane potential. So in the example here, we've got a cell that has a negative charge inside compared to the more positive charge surrounding the cell. And those charges are going to try to move down this electrochemical gradient. And now we've got a situation where we've basically set up the cell as a battery. We've got something that could potentially do work. There's 2 forces at work when we move ions around. Let's say we're taking positively charged ions and we're bringing them into the cell and we're concentrating them inside the cell. Like charges repel, and those ions are going to try to repel each other. They're going to try to get out of the cell. So basically the ions are going to push ions with the same charge across the membrane, except that they can't cross the membrane all that easily. The other thing that's going on is we have a chemical gradient. And that's what we've talked about up to this point. When we talked about things moving down their concentration gradient, we're talking about a chemical gradient. So we have both of these forces that could potentially drive the ions across the membrane. And together, we would refer to this gradient as an electrochemical gradient. So charged molecules or ions are going to diffuse down their electrochemical gradient if they have the opportunity to do so. Electrogenic pumps are pumps that move ions across a membrane in order to set up an electrochemical gradient. And there's lots of pumps that this is what they do. This is all they do. For example, we have proton pumps in the inner membrane of the mitochondria that take hydrogen protons from the interior of the mitochondria and pump them into the space between the inner and outer membrane. And this turns the mitochondria into a battery. What's going to happen is the hydrogen protons are under these 2 forces, chemical and electrical, that are trying to push them back into the middle of the mitochondria. The only place that those hydrogen protons can get across the membrane is to go through something called ATP synthase. And it's really quite fascinating how this works. We will get to it eventually, but the hydrogen protons go through a little channel, and as they do, they cause this almost like an electric motor within the membrane to rotate. And that energy is ultimately used to make ATP. Electrogenic pumps can also be used as a means of setting up an electrochemical gradient that can be used to power other transporters. So here we've got a proton pump that can set up a gradient of hydrogen protons. And now we have something called a sucrose proton co-transporter. The hydrogen protons are going to try to come back into the cell because of that electrochemical gradient. And the only way they can get into the cell is to go through this sucrose proton co-transporter. there's energy contained within that electrochemical gradient. I mean, it takes energy, it takes ATP to set up that gradient, but we can use that gradient, we can harness that gradient to move other things. And this sucrose proton cotransporter uses the energy of that hydrogen proton gradient to move sucrose, and it moves sucrose into the cell along with these hydrogen protons. And we can have several different cotransporters that are co-transporting different things that all run on this hydrogen proton gradient that's being set up by this proton pump. So to sum up, we have simple diffusion, and remember, osmosis is a form of diffusion. We have facilitated diffusion, where molecules are moving down their concentration gradients. Doesn't require any energy, but we've got specialized proteins that make conditions just right for that to occur. And then we've got active transport, where we're using a source of energy, usually ATP, to actively move things across a membrane, and quite often to concentrate things. So far we've been talking about the movement of rather small molecules, but what if you want to move a really big molecule? Or what if you want to move a big chunk of something, like an amoeba taking in a whole bacteria cell? To move big things, we have endocytosis, moving stuff in, and we have exocytosis, moving things out. So exo means outside. Exocytosis can be used to dump out hormones, for instance. Endocytosis, endo means in. This is used for taking in big chunks of cells or whole cells, or taking in large numbers of large molecules as little packets. We have three types of endocytosis. We have phagocytosis, which is cellular eating. Again, this is usually taking in big chunks, so a white blood cell taking in worn out red blood cells, taking in bacteria to destroy them, or taking in debris. We have pinocytosis, which is taking in little pockets of fluid, so taking water, interstitial fluid perhaps, from around the cell, taking it in, and also taking in any proteins and other things that are dissolved in that water. And then we have receptor-mediated endocytosis, which is similar to pinocytosis, except that there's receptors on the cell surface that will grab onto specific things, and the cell will concentrate those as it takes them in. Let's imagine that we have a cell that needs to secrete a lot of hormone, perhaps. So what we've got here is we've got these little packets of the hormone. The hormone would be the red dots. And the little packets of fluid that contain the hormone are surrounded by a sphere of phospholipid, and that's called a vesicle. And that vesicle is going to move to the surface, and it's going to fuse with the plasma membrane and dump the contents out into the outside world. Endocytosis is the exact opposite of that. So dumping things to the outside is exocytosis, bringing things in is endocytosis. So now imagine that the red dots represent something the cell wants. Maybe it's a food item. And what's going to happen is we have a little pocket forming in the membrane, and then that pocket is going to seal off into a vesicle and pull that dissolved food item into the cell. Phagocytosis literally means cellular eating. So we've got an example here where we've got a cell that's producing these pseudopodia. And pseudopodia are little extensions. Literally means false foot. So these two false feet or lobes come off, engulf the food item, form a packet, and then that's brought into the cell as a food vacuole. Now what will happen next is little vesicles that contain digestive enzymes will fuse with that food vacuole, dump the enzymes into the food vacuole so that the enzymes can break down the food. So we've got on the right here a very, very greedy amoeba that is engulfing a bacterium that's almost as big as it is. Your white blood cells are amoeba-like. They produce pseudopodia. They engulf bacteria to kill the bacteria. And in the process, they're quite often killed as well. In pinocytosis, the cell is not taking in a big chunk of something like an entire bacterium cell. Instead, it's taking in a packet of fluid. It's taking in a packet of water, and it's also taking in whatever is dissolved in that water. So let's say that the cell is interested in these little purple triangles. It can take in packets of water. It's going to get lots of that green stuff, whatever that is. Maybe that's something the cell wants as well. But it's taking in a dilute solution of that molecule that it's interested in. Pinocytosis is nonspecific. So when it forms that little water pocket and brings in that fluid, it's getting whatever is in that fluid. Receptor-mediated endocytosis, however, is more specific. So what we've got is, again, we've got a pocket forming and pinching off into a little vesicle, but notice that we have receptors on the surface of the cell. And the receptor is going to bind to that purple triangle. Something that binds to a receptor is called a ligand. So this is going to bind to that ligand, to that molecule. And because we've got these receptors, we are concentrating that particular substance. So notice now that the cell has taken in quite a lot of that purple triangle, whatever it is. The other thing to note is that the pits that form, they're said to be coated. I'm not going to worry about that just yet, but there's motor proteins that make up that coat, and those motor proteins are involved in making that pocket and making that vesicle. So motor proteins are proteins that can change shape. Vesicles are quite important when it comes to moving things around in the cell and sorting proteins. What you're seeing here is the endoplasmic reticulum to the right, and then the Golgi apparatus down the bottom. Proteins that end up in the endoplasmic reticulum can be secreted from the cell. Basically, we have vesicles that bud off of the ER and take those proteins with them, and they may take them to the Golgi where they'll be further modified. In the ER and in the Golgi, that's where we have carbohydrates added to glycoproteins and glycolipids. But in this example here, we have these little green Y-shaped proteins that are stuck to the membrane. And if these are transported in vesicles to the Golgi, they might be further glycosylated, and you can see more sugars being added, so it's more branched now. And then that branched structure can be carried in a vesicle to the surface, to the plasma membrane. And that vesicle will fuse with it, and that glycoprotein will remain stuck to the outside of the cell. We can also have proteins that are just floating around in the ER. If those are shipped off in vesicles that eventually make their way to the plasma membrane, that material will simply be dumped into the outside world. So that's what would happen with a hormone, for instance. So again, those little green structures, those are meant to be integral glycoproteins. They will end up exposed on the surface of the cell, and the little purple spheres there are meant to be soluble proteins. They will be dumped to the surrounding water surrounding the cell. So here we have a cell that's using endocytosis to take in a bacterium. This is what would happen in a white blood cell. Now notice that the bacterium gets taken in, that forms a food vesicle or food vacuole, and that's where digestion is going to occur in the cell. We have a smooth endoplasmic reticulum, and it produces vesicles. It produces lipid. So that's where the phospholipids come from. And those vesicles can move to the surface and dump phospholipid at the surface if the cell has to grow, for instance. But also these little vesicles can carry material around the cell. The vesicles can fuse with the Golgi, and then part of the Golgi can bud off and carry with it enzymes. So enzymes are proteins. They are modified and glycosylated in the rough ER and also in the Golgi body. The lysosome contains these digestive enzymes. It's also got a very, very low pH. It's very acidic inside this lysosome. The lysosome will fuse with the food vacuole, dump the contents into that vacuole so that the enzymes can go to work and break down that food. So the bacterium is going to be broken down into amino acids and carbohydrates and so on, and then those small molecules can diffuse out of that vacuole or be transported out of that vacuole into the cytoplasm so that the cell can use that material. Finally, that food vacuole is going to fuse with the outside of the cell, and any waste that couldn't be digested is going to be dumped by exocytosis. Also note that there are secretory vesicles that are made by the Golgi that can fuse with the surface and dump their contents into the outside world. This whole system of all these internal membranes is known as the endomembranous system. And it's really quite fascinating. It's really fluid, it's very dynamic, and these vesicles are transporting stuff around between different compartments within the cell and transporting stuff to the surface of the cell. And this is just a closer look at how proteins are sorted in the endoplasmic reticulum and in the Golgi. So if you look at the bottom diagram to the left, we've got a ribosome that has docked with one of the sacs that make up the ER. And a protein is being made, and you can see the protein is being passed through a channel into the lumen, or the inner space, of the ER. And that protein can be glycosylated down, it can have sugars added to it. That protein may become embedded in the membrane, and if that happens, it's going to remain attached to the membrane. But now we have a secretory vesicle that's leaving the ER, and it's going to carry that protein to the Golgi where it can be further modified. And as I mentioned before, I was talking about this earlier, we've got two different types of proteins seen in the diagram to the right. We've got soluble proteins, which are going to be dumped out of the cell, and then we have integral proteins that are going to end up attached to the outside of the plasma membrane. Because of this, the membrane has a polarity. And what I mean by that is the outside of the membrane is noticeably different from the inside. And one of the main features, and I've talked about it a few times now, is that we have carbohydrates exposed to the outside world on the outside of the membrane, and we don't see those carbohydrates on the inside. The proteins are arranged to do whatever sort of job it is they have to do. So enzymes that are embedded in a plasma membrane, for instance, quite often they have their active sites on the interior because that's where metabolic activity is happening, inside the cell. With signal transduction, we have receptor sites on the outside of the cell, and then the protein part that sticks into the interior of the cell quite often has an active site that becomes active when something binds to the receptor on the outside. So again, proteins that are embedded in the membrane do a lot of stuff, and they also give polarity to the membrane. Most cells have additional structures outside of the plasma membrane that help protect them. So I mentioned that plants have a cell wall, fungi have a cell wall, animal cells do not, but we have an extracellular matrix. And the extracellular matrix is that kind of layer of protein goo on the outside of the plasma membrane. Lots of collagen, lots of proteoglycans and glycoproteins that help to protect the cell and also help the cell stick to other cells. So in bacteria, we have this special substance called peptidoglycan, which is a mixture of protein and carbohydrate, and that helps support the cell. In fungi, we have something called chitin, which is quite similar to the cellulose we see in plants, except that it contains some nitrogen. The cellulose in plants, as we've talked about, is something you can't digest. Now that doesn't mean it's without any sort of function. That forms the roughage in your food, the fiber in your food. And it's rather important. It stimulates your digestive tract and makes sure that your digestive tract is properly lubricated and covered with mucus. The extracellular matrix is predominantly collagen, and that forms these long rope-like structures, and then also proteoglycans that add to kind of the gel-like nature of the extracellular matrix. Collagen is also very, very abundant in connective tissue. Gelatin, the gelatin that you eat, is boiled up connective tissue. So what they do is they take hooves and bones and so on and boil them, and that extracts the collagen. And here we are with our terminology. Now it is important that you focus on these terms. I've had a number of students ask me if they only have to know what's in the study guide. No, of course not. You have to know everything that's in the PowerPoints. But again, focus on these terms. The study guides just point out some of the big picture stuff, the more obvious stuff. It's a good place to start, but you need to go through the PowerPoints as well.